Good morning, everybody. I think by the third day, people are a little bit exhausted, and they will reach us in due time. Thank you for being here. Our first session today is given by Professor Abdallah Al-Agha. Professor Abdallah Al-Agha is a staff faculty at the Cardiology Department, Cairo University. He is an associate professor of Translational Medicine at the NIH in Bethesda, Maryland, USA. He is a representative of the Euro CMR in Egypt, international. He has several international publications in elite journals like Science and Circulation. His talk today is about translational medicine and how can we serve our community by using this new approach to medicine. Dr. Abdallah, thank you. Thank you.
Thank you, Dr. Yahya, and thank you for Mr. Chairman. Good morning. Today I would like to share with you my little experience in translational medicine, and I hope you find it interesting. So, what does translational medicine come from? When you have an idea for a new treatment or a new device, you just have a methodology and then a research to find your project. But for example, between 1979 and 1983, we had 101 scientific discoveries that were reported that could lead to a medical treatment. However, only 5 received license for clinical use. And believe it or not, just only 1 was a popular treatment. And even when an idea comes through, it takes a long time. So when you publish it in a journal or you have a patent, it takes about from 14 to 44 years to have this project done. The median is 24 years. So we need a tool or a bridge between basic science and clinical science or a product. And that could be represented by what is called translational medicine or translational science. So what is translational medicine? It's considered a process that starts in the lab, what we call it a bench, where a new drug or a new device or procedure is developed, and ends with a production that makes it available in the clinic, which is a bedside. So here's come the name of bench to a bedside. It's relatively actually a new area of investigations that ideally involves the integrated application of innovative technologies in different specialties that encompass multiple disciplines, including physiology, pathophysiology, natural history of disease, genetics, making device, new drugs, and new treatments. And it's also a process wherein scientific discoveries are integrated into a clinical application. So the goal here is to have a clinical product to help the patients. And with these clinical observations are used to generate research foci for basic science. So it's a reversible thing. It's a bench to bedside, but when you have a clinical product, you can go back to the bench just to improve your product. Let's talk about some examples of translational medicine. Since I'm a cardiologist, I'm going to give you some examples from the cardiologist aspect. The first thing, maybe many of you know about the statins or the drugs that lower the cholesterol. And actually, billions of pills have been sold during the last decades. However, we still have some resistance to these drugs, and patients still have high level of cholesterol, especially the LDL. And as you know, we have the good cholesterol and the bad cholesterol. The good cholesterol is called HDL, and the bad cholesterol is called LDL. So an elegant basic investigations performed in a number of labs has shown that there's a protein called PCSK9 binds the LDL at the surface of the hepatocytes and markedly decreased the level of LDL. And now we have this monoclonal antibodies to the PCSK9 that inhibit its function, and the LDL will dramatically be lowered in patients and was proved in clinical trials. The second example is coronary artery disease, and as you know now, it's considered #1 killer worldwide. It's not anymore the cancer, it's heart disease. And usually medical treatment alone is not enough. So many patients, we have to treat them with what is called PCI, or percutaneous coronary intervention, when we put a stent, the metal here that you see here, and we inflate it across this vessel. But however, we make some injury inside the vessel because we are just inflating a metal there. So we were faced by a problem called restenosis. So that vessel will just close again in about 20 to 25% of patients. So the solution was going back to the bench and trying to find just a material that can inhibit the proliferation of cells and lead to restenosis. And now we have what is called drug-eluting stents that release these materials that prevent the restenosis of these stents. There are two phases of translational medicine. The first phase is basic science discoveries that used to develop new treatments for disease. That's called bench to bedside. And the second phase is research aimed at improving utilization of proven therapies in clinical practice and community settings, and that is called bedside to community. And more recently, this has been redefined to include 3 phases instead of 2, with the second phase being subdivided into, so the T1, or the first phase, described the basic science to clinical science, and the second phase, clinical science to clinical practice, and the third phase is used to denote how can we translate this clinical practice to more widespread health improvement. There have been strong international support, and it comes markedly actually from NIH, the National Institute of Health in Bethesda, Maryland. They made a major push to fund translational medicine, especially with the biomedical research, and their focus were on cross-functional collaboration between the basic science and the clinician, and offering new technology and data analysis tools, and increasing the speed at which new treatments reach the patients instead of just waiting 14 or 20 or 40 years. Now it's much easier. And in December 2011, the National Center of Advancing Translational Science was established within the NIH to transform the translational science process so that new treatment and cures for disease can be delivered for patients faster. So if you are new in research and you want just to make a research, what are the process or the developmental process? First, you have to understand the relevant science, develop a prospective treatment, then you develop an assay system. How will you measure your treatment effect? And then you have to test it in a preclinical studies on an animal model, and then begin a formal clinical trial. And if you are in USA, you have to get finally an FDA approval. So creativity is allowing yourself to make mistakes, and art is knowing which ones to keep. And in the following minutes, I'm gonna discuss with you not my mistakes, I hope it's the art, my personal experience in translational medicine at NIH. And this was about a new mechanism in medicine. It's called endothelial mesenchymal transition. Maybe it sounds unfamiliar for many of you, but I'm gonna just describe it, the basic terms now. It was a discovery that of a novel mechanism that could solve the puzzle in cardiovascular disease. So I'm just going to talk about the definition and then its role in cardiac fibrosis and in vein graft remodelings and other applications. So just going back to the basics, you know that all human tissues has 2 cell types, either it's epithelial or mesenchymal, or as the British sounded, mesenchymal, which differ both morphologically and functionally. The basic difference that these epithelial cells are attached to the basement membrane tightly. However, the mesenchymal cells are not attached. The mesenchymal cells are nonpolar, the epithelial cells are polar. So we have different identities of these two cells. The epithelial cells cannot move, cannot migrate. They are attached to the basement membrane. Mesenchymal cells can migrate, can go anywhere into the circulation. And this process, actually, it's reversible. So we can change or transform epithelial cells to mesenchymal cells, and the reversible is true, mesenchymal cells to endothelial cells. So this is a definition of the EMT, where it's an orchestrated series of events in which cell-to-cell and cell extracellular matrix interactions are altered to release endothelial cells from the surrounding tissues. So, excuse me, the cytoskeleton is reorganized to allow movement in three-dimensional in the extracellular matrix. The epithelial mesenchymal transition occurred in the tissues. In the cardiovascular system, we don't have epithelial cells. We just have, instead, endothelial cells. And here comes the term endothelial mesenchymal transition. And it's actually a long pathway upon injury that it's activated, and there are many transcriptional factors that lead to this transformation. One of the signaling pathway is called the TGF-beta. I'm not going to discuss it here. And we have many markers that can help us in identifying this pathway. We have three types of the epithelial mesenchymal transition. One occurs actually in the embryo, where tissues are just transformed from epithelial to mesenchymal. For example, making the heart valves is just epithelial to mesenchymal transition. The second one occurs actually upon injury for tissue regeneration and making fibrosis. And that's the one that we are discussing here. The third type occurs actually in cancer, where cells can be transformed and metastasize. And actually, we got this principle from National Cancer Institute, which is an institute within the NIH, and we have great collaboration between the institutes there. And this is actually a basic thing in many successful universities. So for example, that cancer by itself doesn't kill, but what kills is metastasis. So if you have the cancer just stayed in its location, it will not kill the patient immediately, but what kills is the metastasis. And for order the cancer cells to metastasize, it has to undergo what is called EMT, or epithelial mesenchymal transition. When it's transformed to mesenchymal cells or mesenchymal form, it gets access into the blood, into the circulation, metastasize, go everywhere. When it reaches the target, it has a reversible process called mesenchymal epithelial transitions. And that's why occur the EMT or the metastasis. And when they stop this signaling pathway, they dramatically decrease the number of mortalities in cancer. So this is an example of in the cardiovascular disease that occur actually after heart attack, or what we call it myocardial infarction. After myocardial infarction or heart attack, we have a fibrosis. We have the patients have closed arteries, the muscle doesn't have enough blood, and that will lead to what is called fibrosis. As you see here, this is a muscle, and this is the whitish thing is the fibrotic muscle. And when you have fibrosis in the heart, what will happen? You will have a heart failure, as this patient here. This is an MRI, and show you how the heart is not contracting well. So it's not ejecting blood to the body. So if you have a patient like this, you cannot help him because it's already a fibrosed muscle. So we're trying to understand why fibrosis, what are the source of fibroblast and the fibrosis. It's incompletely understood. There are many hypotheses. One of them, they said it's coming from the bone marrow. These are fibroblasts in the bone marrow. And when the patient has the heart attack, it just migrates to the heart, differentiate into a fibroblast. Other hypothesize that these are peripheral progenitor cells with a cardiovascular potential that may also contribute. Others postulated another hypothesis. They said these are resident fibroblasts within the heart, and they just got activated when the heart attack occurred. So with the availability of different tools that can show the fibrosis in patients, like MRI, that could help us to show the amount of fibrosis. So our hypothesis was whether the endothelial cells within the heart could participate in the injury repair response or simply the fibrosis, and what are the fate of these cells might be. So we have 3 types of cell lineage tracing mice that we used to prove our hypothesis. We simply had induced a myocardial infarction in these mice by closing the artery, the coronary arteries, having infarction, and leaving these mice for 1 month and doing our tools of investigation and then collecting the specimen. These are transgenic mice that are healthy and have normal lifespan, and immunostaining studies, all the endothelial cells will express a YFP, which can give you a yellow or green color. And as you see here, this is a normal heart, there's no infarction, and this is an infarcted heart. And as you see, these mosaic colors of the cells represent the fibroblast. What we have here is staining through 3 different markers. One is for the fibroblast, which is red, and one for the green, which is for the YFP. So if you have a cell that is staining for YFP, which is green, and staining at the same time for the fibroblast, you know that this fibroblast is derived from the endothelium. And we found the number of these cells are increasing during the 30 days. And even in the 3 types of mice groups. And we proved that these are not inflammatory cells. We have to look at the signaling pathway for something called the SMAD2, SMAD3. And we found that the SMAD2 and SMAD3 excuse me, is positive for these infarction cells. What else we done? We took these bone marrow cells from a wild type into a transgenic mice, and we take the bone marrow from transgenic mice into a wild type to prove that the bone marrow is not contributing. It's the endothelial cells are contributing. And what we found that when we put the wild type into a type 2, it doesn't affect the number. However, when we have this transgenic mice into the wild type, the number was decreased. This is the pathology specimen. When you see the blue color, means this is a fibrosis. This is the echocardiography, so we have imaging tools to show the fibrosis by echo and by MRI, which is you see as a white here. That means this is a fibrosis. And the results conclusively demonstrate that these endothelial lineage cells, but not the bone marrow cells, contribute significantly to indigenous fibroblasts following the myocardial infarctions. And these data indicate that EMT may contribute to indigenous tissue repair, raising the possibility that this mechanism represents a new therapeutic target for chemical approach to stem cells repair. We want to proof of evidence, so we looked for the gene and we did a knockout for the gene that is responsible for this pathway, for the TGF-beta and the SMAD2, SMAD3. And when we did that knockout, here's the results. This is the control and this is the knockout. We didn't find any of these cells that were contributing to fibroblasts coming from the endothelium. And actually, the number of the area of fibrosis, this is the difference. The blue color represents the fibrosis, and here's the blue color, is much less. So we were able to decrease the amount of fibrosis in the heart following myocardial infarction, and we proved this by imaging, also by MRI. When you look at this one, this is the control, and this is when you have an infarction, you keep it red. So this is a viable myocardium, it's a surviving myocardium. We also give antibodies to this pathway by called TGF-beta antibodies. We proved the same concept that these cells, fibroblasts, are decreasing in numbers. So we have two unique findings that EMT comprise a large percentage of fibrosis following MI through the TGF-beta pathway, And these results open the door to a new therapeutic approach for endogenously cell-mediated repair, which may be especially relevant for cardiac repair, since we don't have actually a treatment or actual treatment for fibrosis following myocardial infarction. The second thing that we did a vein graft remodeling, and simply, as you know, when a patient has a CABG or a bypass surgery, many of these vein grafts, they took it from the leg, they close after 5 years. Only the artery that they put it to bypass the stenosed vessel will keep patent. So this is actually a major problem for cardiac surgery. And we try to understand the mechanism why these vein grafts just close. So there's many hypotheses for this, but the classical response to injury that will happen, endothelial cell damage will occur, growth factor release, and smooth muscle cell proliferation, and then migration to form the new intima. Our hypothesis was different, that EMT, our endothelial mesenchymal transitions, may be involved in the new intimal hyperplastic response, which occurs after vein grafting into the arterial circulation. By the same concept, we have a transgenic mice, and we took a jugular vein from this transgenic, from a donor animal, and attached it to a femoral artery of the recipient by end-to-end anastomosis. This is a femoral artery that we took a vein and put it into an artery, and now it's exposed to arterial circulation, high blood pressure, and that will induce injury into the vein. And we did also an endothelial lineage tracking during the vein graft remodeling. And here's the evidence, or the proof of evidence. When we gave the antibodies to the TGF-beta antibodies, here's the TGF-beta, and this is the control. You see this is a cut section of the vein. You see that this is the lumen, just the small thing here is the lumen, and this is wide patent vein graft. So we were able for the first time to keep a vein graft patent after a surgery by understanding the molecular mechanism behind this. Also, we have to prove it by genetic knockdown, by knowing that the gene, the SMAD3, and do a knockdown. And same mechanism, we found that when we stop the gene acting on this endothelial mesenchymal transition, we keep the vein patent instead of having proliferating smooth muscle cells. We have other applications for, in the literature for epithelial or endothelial mesenchymal transitions in the lung, in the pulmonary hypertension, in the liver, when they treat cirrhosis, for the kidney, for the chronic fibrosing type injuries, and for other organs. So it's a promising mechanism that can be used in different entities of medicines. Also, it has been used into in the cornea, in treatment of corneal opacities. And moreover, now there are some studies on the application of this EMT, stopping this mechanism to treat atherosclerosis in the vessels, and all the vessels of the body. So my conclusion is that translational medicine is a rapidly growing discipline in biomedical research and aims to expedite the discovery of new diagnostic tools and treatments by using a multidisciplinary, highly collaborative bench-to-bedside approach. And EMT, or endothelial mesenchymal transition research, in the next few years promises to be exciting as new mouse models and molecular probes are identified to address important and still unanswered questions. Thank you very much for your attention.
Thank you for beautiful research and a very nice presentation. I particularly enjoyed it very much. If any person in the audience has a question. They understand it very well, so no question.
Thank you, Doctor, for the great lecture. I want to ask, can we use the same concept to prevent the squamous cell metaplasia in case of cancer?
Excuse me, to prevent what?
Metaplasia in case of cancer.
Oh, actually, there's a at NCI or in the cancer field, they just they have a great results in combining different stages of cancer and even metaplasia. There are some studies on that, but actually billions are just paid for NCI just to prevent the cancer, and they showed that the mortality just decreased in cancer cells. They have a great celebration, I think it was in 2010 at NIH, and President Obama came and he gave actually, during the crisis, he gave more money to the NIH just for the NCI because they have good results in combating the mortalities of cancer. And this is actually one of the great things in history of medicine, is to decrease the number of cancer mortalities. Now it's not like before, if you have cancer that you're dead. No, it's different. And metaplasia, as you said, it's one of these stages that they can actually help it in early stages of the cancer spread. So you need to just to know when to stop this cancer, but also you need good imaging tools just to know that this patient. So you need translational medicine because it's a collaboration between clinician, basic science, imaging tools. All of these will help to improve the health of the community.
It's a very nice presentation, but just to check my understanding. So now you want to prevent the progression of fibrosis, not to fix or to return back the fibrotic tissues into a viable and just to stop the progression of the disease by blocking the SMED2 pathway.
Exactly. We don't want to prevent all fibrosis. We just identifying the mechanism, because if you block all the fibrosis, the heart will rupture. So this is a good mechanism. We have fibrosis in the body is made just to protect cells after, or tissues after injuries. So you just replace these tissues, the dead tissues, by fibrosis. But you don't want excessive fibrosis. That's what the body made the mistake. It's an excessive fibrosis. So as I show you, the patient will get a heart failure. So we want to have just a balance between fibrosis and viable tissues. And this is very important just to know the mechanism. Before that, we didn't know the mechanism of fibrosis. We don't know where are the fibroblasts coming from. So now we have an evidence. These are derived from the endothelial cells. And endothelial cells mean vessels. And the vessels are rich in all organs. So every organ has endothelial cells, especially the heart and the arteries.
I want to ask about the association between the genetic polymorphism of genetic factors such as SMAD3 genetic polymorphism, TGF-beta1 genetic polymorphism, as a genetic marker for susceptibility of myocardial infarction. Okay.
Actually, we haven't studied this. We just know that this is, we just identified the pathways. So we haven't studied the polymorphism of this genetic pathway. Actually, it's a good question, and maybe it's a good research in the future.
I am considered that a genetic marker for progression of myocardial infarction as interindividual variation between the genetic polymorphism of this gene as a risk factor. Okay.
I understand your question, but here this will represent just the amount of fibrosis that will happen. But to predict myocardial infarction is completely a different story. It's other risk factor. If you have high blood pressure, hypertension, high blood sugar, high lipids.
I understand.
It's different things. So you cannot just predict. You can maybe predict that this patient will have an excessive amount of fibrosis, or will not have any amount of fibrosis, or if you have a polymorphism. So some patients actually, after myocardial infarction, they died immediately. Why? Because they don't develop fibrosis, because the heart rupture. So maybe these are small entities of patients that have some problems in having fibrosis. So we want to reach a balance.
Genetic susceptibility.
Yes.
genetic susceptibility of myocardial infarction.
Exactly.
Thank you.
One question. Only we have time for one question. We have to
Yes.
move on. Professor Agar will be available for networking after the session. So your last question.
Okay, thank you very much. Thank you for your presentation. Could you just clarify, in your case, how bench to bedside will be made shorter? Does it involve multidisciplinary sort of centers studying the same problem? How will it be made shorter to reach the patient?
Make it shorter by first finding the product that we have an antibodies or a molecule that can stop this TGF-beta pathway, or specifically the biomarkers, the SMAD2 or SMAD3. We don't want just to stop the whole TGF-beta pathway, just a specific molecule. And if you stop this molecule, you will be able to decrease the amount of fibrosis. So how it's make it faster? By having the cardiologist, the interventionalist who will do this, by the geneticist, by the basic labs personnel, all of these, when they have a cooperation, will make it shorter. We will just increase the survival rate after myocardial infarction or decrease the complication of heart failure. We have many patients with heart failure, and unfortunately, we cannot do anything. And like maybe 50% or 60% after myocardial infarction have heart failure. Thank you.
Thank you very much. And I now call on Professor Heba Asim, who is the Director of Clinical Genomics Center at the Alexandria University New Hospital. Heba is an assistant professor at the Medical Genetics and Executive Director of Clinical Genomics Center at the Alexandria Faculty of Medicine. She also acts as a medical genetic consultant in Dr. Magdi Yaoub Heart Foundation and co-investigation on a national international research program investigating the genetics of cardiomyopathies in Egypt, particularly the congenital one. She has attained her PhD at Manchester University in UK and has an MD in molecular biology in the pathology department since 2004, Alexandria University. Her special interest in clinical molecular genetics from a diagnostic and prognostic perspective. She has special interest in cancer and cardiovascular hereditary diseases and provides clinical care and genetic counseling to families with inherited disease. She has several international publications on the subject. Please.
Thank you very much, Professor Yahya Halim, for this very kind introduction. And I would like to thank the organizers for this wonderful meeting. I very much appreciate your effort and support to all the participants. Thank you very much for your effort. It's a real pleasure and honor to stand here in front of you and to talk to you about the applications of the genomics in medicine and how the development of technology has added value to this branch of medicine. Now, the content of my talk would be what is meant by the recently used term of functional genomics. What are the key features of this developing science? The use of this for genotype-phenotype correlation, and to apply this, I'm going to give a disease model, and that is the hypertrophic cardiomyopathy, the genetics of hypertrophic cardiomyopathy, and the application in the management. And this would be the example of the application of translational genomic medicine that can be applied to other disease disorders. So, functional genomics is actually what comes following the identification of the genetic defect, following the knowing of the mutation that is there. And what follows this is the effect on the transcription, translation, regulation of gene expression, and the protein interaction. So the first part of the science is static, when you identify the mutation. This is a permanent, heritable change. And then what follows is the dynamic genomics that maybe you can manipulate and change the outcome. So the key features of this science really is the high throughput analysis rather than gene by gene analysis, and the genome-wide approaches, which we have heard a lot about that yesterday in Dr. Ahmed Mustafa's talk and Dr. Rami Aziz's talk, and you can see that it applies to all organisms and all species. And then the value behind this is the correlation between the genomes of the organisms and the phenotype. And in medicine, on a lesser scale, we want to do a genotype-phenotype correlation. So, our scientific promise working with functional genomics is integration between genomics and proteomics. Why? To understand system biology. Why? To identify phenotypes, be it that phenotype is normal or a disease. And this was very nicely elaborated in the talk of Dr. Sharif Al-Khamisi yesterday when he was talking about the spinocerebellar ataxia, another disease model, and how and he wonderfully showed us how stepwise the analysis has taken us to the linking of the gene with the disease. So the technical progress of genomics analysis is really like any experiment you do in the lab. You take your sample, you do the experiment, and then you interpret the results. Well, in genomic research now, you do a biological experiment, maybe just by watching the phenotype of the patient, but currently, in the omics era, you are doing really high throughput technologies. And not only that, we have now a new science emerging, and that is a transdisciplinary bioinformatic analysis. And then you are flooded with data, a lot of results that you have to interpret in context of the gene, the transcripts, and the protein. So let's then now work in functional genomics. You have three main entities to work on. DNA, where only 2% of it is representing the coding sequence of your genes. Of the known genes so far, you have the RNA and you have the protein. Now, these three principles represent the principal equation of life that we call the central dogma of life. So your approach can be by a single gene approach in the DNA, using DNA, or a targeted panel approach where you take panoramic view of gene panels that you have is evidence of its involvement in the disease of interest, or whole exome sequencing, or whole genome sequencing at the moment. And with the RNA, you can study the transcriptomics of a certain cell or tissue, maybe the microarray, which can show you the expression profile. And recently also there is the emerging microRNA sequencing, which is going really to revolutionize functional genomics. And then, of course, you have to put this into meaning, into functional molecule meaning, and that is by working on the protein. And this is through the protein-protein interaction, undertaking the functional assays through animal models and in vitro assays, and maybe also biological assays. And, of course, you have the knockout mice, transgenic mice, which can show you the disease in front of you in short of duration of life of the mouse. Now, these all are actually interrelated, and one can make you understand the other and link them together. So you will be facing enormous amount of data, and you have to do computational bioinformatic analysis, and not only this, but also modeling. And here comes the role of the biomedical engineering, which is emerging and flourishing these days. And these are all done in silico analysis. Now why are we doing all this? Well, we want to know biological translational meaning for what we are doing in the lab. So, in the lab, you're going to identify a mutation, and it doesn't end here. There is a lot of intellectual work that you have to undertake, and this is to assess the influence of this change on the function of the protein you are studying, on the is it related to the quantity, and that is why its deficiency is the cause of the disease? And then you can evaluate its pathogenicity. So really you have to be determining, is this mutation causing a loss or a gain of function? You really need to weigh this out in order to link it with the pathogenesis of your disease of interest. We all know that the protein structure starts from a primary structure, that is the plain sequence of amino acids, taking this into the secondary structure where parts of it is helical, parts of it are sheets, and then to a tertiary structure with the conformation of the protein molecule in order to divide it into functional units, each performing a particular function. And with some proteins also, they only act when they form multimeric molecules. So this is the secondary structure. You can see here in this part, there is the sort of the sheets forming part of the protein, and other parts are forming helical. Now this all depends on the interaction of the sequence of amino acids with the polarity and the mass actually of these amino acids and their interaction together, giving us ultimately the tertiary structure, which is the actual functional molecule interacting with its surroundings. So in order to determine the pathogenicity of a mutation, you really have to go through a checklist, if you like. Going from the strongest, that is the functional studies. It could be in vitro assays using cells, or it can be through the IPS, or it can be through animal models, and this can easily be done in the zebrafish and in the mouse models. Or this mutation can be previously described in relation to this disease of interest, or it might be a de novo mutation detected only in the affected patient but absent in his parents who are normal, or sometimes you are faced with a novel sequence change. In such case, you really need to exclude it from 200 healthy controls. They have to be healthy controls, not having the disease of interest or even other known diseases. And this has usually been, or according to the molecular guidelines, diagnostic guidelines, have so far been screening in 400 alleles, that is in 200 subjects. Now with the technology of the NGS, this has really escalated to like 1,000 controls. And they should be ethnic matched. And this was what Yasmin Hagib was saying, is that currently we are looking at our Egyptian population with NGS to determine these novel rare variants. Are they rare variants specific to our population, or they are only present when the disease is present? And then of least strength is to look into the chemistry and the nature of the change and do the in silico analysis and the modeling work. So we come to why do we do this? We want to establish a genotype-phenotype correlation. It should be easy. A gene produces a protein, and this protein produces a phenotype. Very simple, straightforward relationship. And especially so with the single gene diseases, single gene change can be directly related to clinical phenotype. Unfortunately, this is not the case with all diseases, and we get diseases with a phenotype heterogeneity, where you can have the same mutation in the same family with a wide, broad spectrum of a phenotype. What is happening here, that is the effect of the surrounding environment of the genes, which act as modifying gene effect, or environment effect. So it's not straightforward. Now I move on to my second part of talk, which is the application of this basic science and basic knowledge in functional genomics and in medicine, the field where I belong to. Well, with every exploration, like what Magellan has done in the early 16th century, he did a circumnavigation of the globe, starting from the east and going back to the east across the Pacific Ocean, which he called the Sea of Peace. Following this exploration or circumnavigation, a lot of land has been discovered, and exploitation of its treasure is still ongoing till this very moment. So the same thing happened really with the Human Genome Project. The Human Genome Project that has finished in the early 20th century is going to be exploited for years to come. And interestingly, the Human Genome Project for the sequencing of the human genome costed at the time $400 million. Now today, as we have heard yesterday from Amr Aziz and Ahmed Mustafa, a whole genome sequencing is undertaken in a single lab by a single researcher and is costing $1,000. So you can imagine what we can do. Now I will talk about the disease model, and I will start with this slide of the review of Sir Magdi Yaqub in Nature Genetics, in Nature Reviews. And he was talking about the genetics of cardiomyopathies. And this diagram beautifully correlates the different types of the phenotypes of the cardiomyopathies. That is the hypertrophic, where you get thickness of the heart. That is the dilated, when you have thinning out of the heart. And that is the arrhythmogenic right ventricular cardiomyopathy, where you get fibrosis in the subendocardium and arrhythmias. And you get and there is the fourth type, which is the non-compaction cardiomyopathy, where there is persistent of non-compaction pattern of the myocardium as the fetal heart. And these have been linked to families of genes. So you have the hypertrophic cardiomyopathy, mostly linked to the sarcomeric genes, and you have, for example, the arrhythmogenic right ventricular cardiomyopathy linked to the desmosomal genes and possibly also the ion channels. And you have the non-compaction, mainly concerned really with developmental pathway of genes like the Notch pathway. Well, when we started the Egyptian HCM national program that started back from 2007, it was only wise to start with the 3 common genes that have been reported to be commonly involved in HCM in other populations. And these are the myosin binding protein C, the myosin heavy chain, and the troponin T. And these are the results of the samples so far analyzed from the time of start of recruitment back in 2007 till the end of 2015. And the early results were reported in 2013 in the Journal of Cardiovascular Translational Research. Well, with these 3 sarcomeric genes, we were able to detect pathogenic or a likely pathogenic mutation in 50% of our patients. And myosin binding protein C scored as the commonest gene, followed by myosin heavy chain in 20% of patients. The cardiac troponin T, which is a regulator troponin T, is involved in like 4% of the cases, and we had some patients who had double hits in like 2% of the cases. We would expect those with the double hits to have really a more severe phenotype than others. And still we are left with 50% of our patients who have a clinical phenotype of idiopathic cardiomyopathy, but with unknown genetic defect. So came the era of the next generation sequencing, and through Sir Magdi Yaqub, he kindly established the collaboration with the cardiovascular genetics department at Imperial College. So we did a pilot study on 150 patients who were co-analyzed in our lab here for the 3 sarcomeric genes, together with the NGS for a panel of 150, for a panel of about 150 genes which are involved in inherited cardiac conditions in general. And our colleagues there and collaborators in the genetics lab did the experiment of the library construction and the bioinformatic analysis using their set-up pipe stem analysis. And as Yasmin has mentioned yesterday also, now an Egyptian scientist has went there to study this and to do it by the Egyptian hands and to transfer the technology, inshallah. And these are the results that came up from this over 100 genes. Again, myosin binding protein C scored commonest, followed by myosin heavy chain, cardiac troponin T with other sarcomeric genes. But the interesting thing is that the patients who had multiple hits with either sarcomeric genes or non-sarcomeric genes were escalated by like 10 times. So they were like 22% of the cohort. And other interesting findings really come up. Now these results are very new, and the publication is still under construction. So really, it should remain in within this hold. And we were able to compare our analysis in the past using the denaturing high-performance liquid chromatography for detection of heteroduplex, because this disease is autosomal dominant and it is expected to be present in a heterozygous status. And the variants that we detect by this high-throughput analysis for screening will be confirmed by a Sanger sequencing. Our results on these 150 patients was compared to the results obtained by the NGS, and again, the variants were reconfirmed by Sanger. So we considered the Sanger as our baseline of analysis. So you can see here that the variant detection rate in the 3 genes that were co-analyzed with the 2 methodology has jumped up by 10%. And another interesting piece of information was that we detected homozygous individual in this autosomal dominant disease in 6 of our patients, patients. Now that is like 4% of this cohort. And this is very high in comparison to other populations. But this is not strange for the Egyptian population, knowing that the consanguinity rate is reported to be between 25% and 30% just by society traditions. So the sort of sensitivity specificity comparison, we found that for the analysis that we were doing with the DHPLC, it was like 90%. And this is really because of the homozygous samples. You can see here that in this graph here, this electropherograph, you cannot detect, this is the wild variant, and amongst them is a very thin white line. I don't think I'm pointing at it. It's in the middle, in between the wild variants, and this is the homozygous sample. Thank you. And here, there is a heterozygous sample as well that was not picked up, and it had the profile of the wild type. So the sensitivity of the DHPLC was missing some samples, and the reported actually sensitivity is 95%. It was higher in our cohort because of the homozygous samples, which are not likely to be detected with DHPLC. And then the results were repeated after the ICSAC database was established. I worked with my friend Roddy Walsh from Imperial College and asked him to do the reanalyze the samples after the ICSAC database was established. And kindly, he did this, and this is the graph that came out, and it shows that the sarcomeric genes are the most commonly involved in our Egyptian pilot, and the other sarcomeric genes are present, but to a lesser extent. But the interesting thing is that we had calcium and sodium channel ions involved also in hypertrophic cardiomyopathy. And there isn't much in the literature really linking the ion channels with hypertrophic cardiomyopathy. So this really needs more investigation and working out. So we undertake our biological functional experiment, looking back at the family, and luckily in this family, here is our index case. She's a female, 23 years old. She had also an affected brother and an affected father. So we looked for the variant present in the sodium channel ion gene, SCN5A, and we did find that the mutation was co-segregating with the phenotype in the brother and in the father. Now this is a nice biological functional test. Just by looking at the family, we find that the variant is correlating with the phenotype. I come to the third and final part of my presentation, and that is the translation of this knowledge in life, in management of hypertrophic cardiomyopathy. And in 2011, the expert consensus came up for the role of genetics in inherited cardiac diseases, the channelopathies and the cardiomyopathies. And they have decided for hypertrophic cardiomyopathy that genetic testing for an index case who is clinically diagnosed with hypertrophic cardiomyopathy to undertake genetic testing, particularly for the sarcomatic gene panel, is considered as a class 1 recommendation. And also the analysis I'll be on time, thank you very much. And also for the family members, once a mutation is found, is also a class 1 recommendation. Now, we are going to be diagnosing family members who do not have the disease with a genetic mutation. So this would be bad, sad news for healthy individuals in a family where a mutation is segregating. Well, not really. It will actually be good news because for this category of people who are genotype positive, phenotype negative, we can do something to modify the risk of development of hypertrophic cardiomyopathy, and the disease phenotype can be modified and can be milder and perhaps delay its onset, or it may not even happen by taking a calcium channel blocker. Now, this is the very early results of work done on use of calcium channel blockers as a preventive treatment for the genotype positive, phenotype negative family members belonging to a family where a case is diagnosed with hypertrophic cardiomyopathy. And this is actually the value of translational research. And as my colleague, Professor Al-Agha was talking, translational research is really the goal for us clinicians who care for patients and transfer the knowledge from the bench to the bed. Now, I must acknowledge and heartily thank the King of Hearts, Sir Magdi Yaqub, for transferring this ad hoc research, high-tech research, to be undertaken here on the land of Egypt. I also thank the team in Aswan Heart Center, and Yasmin has shown the structure of the research department and how it integrates with the practice of medicine there. I have also to thank the Bibliotheca Alexandrina, and here amongst us, the chair of this session has been involved heavily in the establishment of the MOU back in 2006 or even earlier when I did, maybe before I was here back from England, for doing this research under the auspice of Bibliotheca Alexandrina in the BA MYF Genetics Lab. I have to thank also the young scientists, hardworking, dedicated Bisra Sami and Rimon Sobhi. Please stand up and please give them a round of applause for their hard work and dedication during this program. Of course, I thank you very much for your work. I also thank the national cardiology team, the satellite clinics present almost everywhere now on the map of Egypt. I heartily thank the Egyptian patients and the healthy volunteers for their kind patience and their heartily participation, altruistic participation in this program. They have worked with us since we started, and they would wait for years for the results to come out. It's not easy, and people who are working in the lab, and especially working in the sequencing at a scale of a gene by gene analysis, does know how much time and effort it takes to get out results. And of course, I thank our international friends and collaborators from Imperial College, Carigi Hospital in Florence, and earlier as well from Oxford University, who have helped us in establishing the BA-MYF Genetics Lab. I haven't put any names because I didn't want to miss any names. Hundreds of people are working on this program, and the intellectual ownership is reserved for everyone through the list of publications that are present and will be present soon in the future, inshallah. I wish to thank you all, and really geneticists work with clinicians in different specialities in order to give high quality medicine. Thank you very much for your kind listening.
Thank you, Heba, for your valuable contribution, and I've been following that for a long time. We have only one question because we will be running out of time, and there will be no time for coffee, which is a very important item in our daily work. Now,
first of all, many thanks for your interesting lectures. I just would like, or I'm interested to know, what is your interpretation that people who already carry heterozygous in particular positions of myosin-binding protein are more susceptible to cardiomyopathy than the other people who already carry homozygous nucleotide?
It's not that they are more susceptible, it's the pathogenesis of the disease. One mutation in one allele, of course you know that we have two alleles for each gene. So one is enough to cause the disease. That is what is known about hypertrophic cardiomyopathy. But when you have two hits, now this is likely really to be lethal, to cause severe, very severe phenotype. And we're not observing this in all our homozygous patients. We find like almost a similar clinical picture between the homozygote and the heterozygote. And this is really strange, and it is worth further looking into.
thank you, Professor Heba. Thank you for your excellent talk. Our next speaker is Professor Sameh Ali, who is a professor of biophysics and founding director, Center of Aging and Associated Diseases at the Wales City of Science and Technology. Now, biophysics is a very important field in any medical faculty or any medical basic research. And I personally believe that if you do not have proper biophysics in your institution, research will be extremely difficult because it has opened the way to a lot of fields and brought all disciplines together. So Professor Sameh returned from California, San Diego, in June 2012 to participate in the establishment of one of Egypt's national projects. Dr. Ali is formerly trained physical chemist, but his interest in the free radical biology and medicine started in 2003 while exploring the extraordinary antioxidant properties of fullerenes in vitro and in vivo, the elegance and complexity of mitochondria, the tiny powerhouse within our cells that was once a free-living bacteria, fascinated him so much that he made a career shift to study the critical roles of mitochondria in aging and diseases. Dr. Sameh, floor is yours.
Thank you so much, Dr. Yahya. Good morning, everyone, and I'm really honored to be among the speakers of this, what is becoming a very, very important meeting and a very good opportunity for everyone to interact with prominent scientists from all over the world, and also with young researchers, promising researchers in this country. Today, I actually decided to take you a little bit in a different direction than what we've been hearing today and yesterday, translational medicine. I'm probably going to step down a little bit and go with the initial phases of what's so-called translational medicine. We have a preclinical study, and I decided to share with you the excitement that we have. But of course, depending on the culture, if you say in your talk that you have some bad news and some good news, and then you explore whether to start with the good news or bad news, I personally select to start with the bad news, which is reporting on causes of death in Egypt, and it's also reporting on the severe health-associated problems in Egyptian populations. As you can see, we are the first globally in terms of the rate of infection and rate of diseases such as bladder cancer. We are also the first in hepatitis C infections. We are number 3 in terms of endocrine disorders, and also we are number 3 in terms of hepatitis B, and so on. It's a very heavy packet that anyone who is interested in biomedical research and also in the clinical applications of biomedical research is always thinking about this. And this is exactly why I decided to focus primarily on mitochondria. Mitochondria is very well known as the powerhouse of the cell. However, recent findings is exploring and is confirming that mitochondria's role in the cell biology, diseases, pathology, and even in physiology is beyond being the powerhouse of the cell. Mitochondria is or are important elements in signaling, and particularly or of particular relevance to today's talk is very important element in induction of apoptosis. Of course, we will talk more about this and in the later parts of the presentation, but this is why we have decided to focus on mitochondria research in Zuwail City, where we established the Center for Aging and Associated Disease, because of the central and very fundamental contribution in the biology of so many disorders, including neurodegenerative diseases, endocrine problems such as diabetes, also cardiovascular diseases and aging, which is important to everyone, evidently. And in particular also, the new trend of the importance of mitochondria in cancer research is also highlighted in this talk. So since mitochondria and mitochondria studies are not really common, especially in Egypt, I always start by discussing or introducing how to study mitochondria. Mitochondria are very tricky organelles to study, but of course, since they are responsible for more than 95% consumption of the oxygen that we breathe, definitely respiration is very important. And to study respiration, you need to study oxygen consumption under certain conditions. You need also to see if this oxygen is transformed into the cellular kinds of bioenergetics, which is ATP. And also, you should also study the different metabolic pathways. Many of these metabolic pathways rely on mitochondria, but some of them are not, such as glycolysis. And glycolysis, by the way, is very important in cancer research, as we will explain a little later. One of the elements that mitochondria is contributing to pathologies is the production of reactive oxygen species. These are very reactive intermediates that are known to cause random damages to cellular components, macromolecules, including DNA, including cellular membranes, including proteins. And the current consensus is that reactive oxygen species are very important in the aging and in neurodegenerative diseases, while being utilized or exploited in the biology of cancer. So cancer is essentially exhibiting a sort of mild oxidative stress. This would help cancer cells to proliferate and to survive harsh conditions. So we are also interested in studying how much reactive oxygen species are produced, when are they produced, where are they produced, and the type of damage that they inflict in cellular components. Again, important aspects of mitochondria studies is the apoptotic mechanism, also the contributions of calcium dynamics, the role of mitochondria as thermogenic elements in the cell. We also are interested in the structure of mitochondria and how it correlates with the function of mitochondria. And then the dynamics, which is a terminology that's expressing how mitochondria is undergoing fission and fusion. And to study this thing, we established a laboratory in Zuwail City where we have a cutting-edge instrumentations where we can study mitochondria function and metabolism in great details, including also reactive oxygen species. And this is one of our main horses to study mitochondria. This is a system that's based on electrochemical measurements in addition to also fluorescence measurement to measure high-resolution oxygen consumptions. And this system would essentially enable you, with all the accessories that it has, to study any sort of cellular or any sort of biological specimens, including tissue homogeneous, including cell suspensions, including also adherent cells. And for example, you can talk about how this system is really changing the biology of mitochondria because of the very long-term stability and linearity of this measurement techniques. I'm not going to discuss a lot about this, but it's actually one of the advantages is that it actually can reach picomole sensitivity, which would enable you to study very, very tiny and little amounts of mitochondria or isolated mitochondria or cells. For example, you can use this system to measure the maximal respiratory capacity of a muscle, which is a very important aspect in muscle performance and muscle function. And you can, in parallel also, this is an example, for example, how the heart is changing in its metabolism or its rate of oxygen consumption. And later on, we did many studies to see also how this is associated with reactive oxygen species production, because mitochondria are suspected to be one of the primary sources of reactive oxygen species in the cell. You can also study the maximum or the maximal respiratory capacity, which tells you how your organ is capable of handling stress. So a higher respiratory capacity or reserve capacity indicates that your organ is capable of handling stressful conditions and demands. As I mentioned, also this system is equipped with many other accessories that would enable you to study hydrogen peroxide production, to study mitochondrial membrane potential, to study calcium, and so on. And in parallel, at each respiratory state that you stimulate, you can measure hydrogen peroxide production, which is very important to know exactly what are the percentage of reactive oxygen species that are produced when you are consuming oxygen. The other system that we are also using is what's called seahorse biosystem. This is quite important in the story of cancer that I'm going to talk about. Of course, metabolism is extremely complex, but we focus on two major sources or two major pathways of metabolism in cancer cells. This is a glycolysis, which produces lactic acid and measured through the acquisition of extracellular protein release due to lactic acid release from the cell, in parallel with also measuring the mitochondria-mediated oxygen consumption. This system is a very nice technological advancement which would enable you to study a monolayer of adherent cells in a very, very tiny volume, which can reach down to about 6 microliters, which can enhance the sensitivity and bring it down to changes in the pH value, milli pH values, and also in picomole oxygen consumption. And to be able to handle or to intervene with metabolism in cancer cells or any type of cells, you start by profiling your cells or profiling your organ. And how you do this is essentially you measure both the oxygen consumption rate due to mitochondria, and you also measure in the same time, you measure extracellular protein release, and you plot a specific relationship between these. If you have the relationship is on the lower bottom on the right hand, this means this is a system that's relying more on glycolytic activity. If you have something like this, if you can see both of them, this means that there is an exchange or interaction between both pathways, and the cells are relying on both. If you have higher oxygen consumption, it indicates a sort of reliance on fatty acid oxidation through mitochondria. If it is in the quadrant to the upper right, it indicates that your mitochondria is uncoupled. And this is very important, for example, in thermogenesis, or if mitochondria is poisoned, for instance. So this is what you essentially do. You profile mitochondria, and you try to first identify, or you profile your cells to identify the type of metabolic pathways that are important in each cell. Then when you apply this, for example, we are interested in this research on targeting cancer in liver cancer, hepatocellular carcinoma. And as you see, this is a very, very important health problem in Egypt due to the very high rate of hepatitis C infections. And in general, if you just go through the causes of cancers in general, you would find that smoking and also the unbalanced diet are very important. So this hints at the role of metabolism in induction of cancer. If you move forward to just to talk about what are the hallmarks of cancer and how to tackle this, first of all, you would see that any cancer is evading apoptosis. There is a way that cancer cells are essentially avoiding the apoptotic responses, and this is, of course, primarily mediated by silencing mitochondrial contribution in the cell biology. There is also self-sufficiency in growth signals, and there is also an evasion of anti-growth signals. Other hallmarks of cancers include limitless replication, sustained angiogenesis, evading immune surveillance, DNA damage, tissue invasion and metastasis, and oxidative stress, as I mentioned, is a sort of a hallmark of cancer that you can see that in most cancer cells you have a persistent oxidative stress, mild oxidative stress level. And finally, we have metabolic changes. And these last two hallmarks are the subject where we essentially will try to draft or try to devise a strategy to handle cancers. So the hypothesis is that in cancer cells, there will be some sort of altered metabolism. And this is probably resulting from limited supply of oxygen or the fundamental way of trying to avoid the contribution of mitochondria in the apoptotic responses in cancer cells. And there will be, to compensate for this deficiency in mitochondria metabolism, there is the enhanced glucose uptake. And essentially this leads to a sort of persistent oxidative stress. You will see that this is present in most cancer cells, and this would lead to uncontrolled proliferation and metastasis, and resulting or mediated by DNA damage. So our hypothesis is that what would happen if we try to intervene with metabolic changes that are present in cancer cells? For example, if there are some reliance on glycolysis, what would happen if we particularly inhibit glycolysis? And if mitochondria is there, what would happen if we activate mitochondria pathways to see how this would affect the survival of cancer cells. Metabolism is very, very complex phenomena, and as you can see, it's very intricate. And if you don't believe me, this picture probably is very, very complicated. But if you don't believe that it's really simplified, this is another picture of the metabolic pathways. But we are essentially focusing on two major pathways: the glycolysis and the mitochondria-mediated oxidative phosphorylation. So in quiescent cells, normal cells, what happens is usually glucose metabolism enters through the transformation into pyruvic acid or pyruvate and enters the TCA cycle to produce ATP and to break glucose completely into carbon dioxide and water. There is some contribution from fatty acids, some contributions from glutamine, depending on the state of proliferation of the cell. But in highly proliferating cells, what happens is that there is a huge reliance on glucose, where glucose is utilized to produce biomass. And there is some sort of also reliance on mitochondria through the TCA cycle. But the primary utilization of all of these metabolic pathways is to produce biomass. In cancer cells, or in normal cells, let's start with that, what would happen if you are challenging your cells, immediately glycolysis comes into play. But gradually, you cannot afford continuing to use or to utilize glycolysis because this produces a lot of acidosis, and this would lead to cell death. So mitochondria would kick in gradually and would continue to supply the needs of a cell. However, in cancer cells, what happens is that oxidative phosphorylation or mitochondria are usually low in contributions, while glycolysis continues to supply bioenergetic needs in a cell. So what will happen? The question is, what would happen if we essentially try to do a metabolic switch? Metabolic switching is an important strategy, and it's not really very new. It's started maybe about a decade ago, and it was motivated by several facts that oncogenic signaling and tumor metabolism are very, very interconnected. And malignant transformations by various oncogenes, you can see there is a very, very important connection between tumor suppressors. Most of the mitochondrial genes are tumor suppressors. And you can see in cancer cells there are some sort of avoidance of mitochondria for that reason. So what we are attempting to do is to test a hypothesis whether metabolism is really the Achilles heel for cancer cells, and this is through, of course, drug screening to find agents that can essentially, in a controlled way, target a specific metabolic pathway. So in any cancer, this is a very general slide. In any cancer treatments, what you actually are after is you need to target cancer cells while sparing normal ones. You need to target molecular pathways that are characteristic of cancer cells. And you need also, or you may be looking or seeking a targeted delivery so that you avoid the targeting of a normal cell. And then you try to minimize the effective dose of your drugs. And you try to induce apoptosis or autophagic cell death rather than necrotic cell death, because there are a lot of therapies that would probably that can actually induce necrotic cell death, but necrotic cell death is inflammatory in its essence. So a potential strategy is to do what's so-called metabolic switching strategy. This is essentially, I like to call this just like the 007 strategy, where you surgically target a specific metabolic pathway and you try to switch this metabolic pathway. Like, for example, what we try to do is to inhibit glycolysis and activate mitochondria and try to see what happens. There is the other approach, which is metabolic blackout strategy. You are now targeting all metabolic pathways, and I call this the Terminator approach. So we screened a library of drugs to try to find some of these drugs, and essentially we arrived at certain formulations and certain chemical compounds that can inhibit glycolysis while activating mitochondria, as you can see. The advantage of this is that you can specifically target cancer cells, and there is no expected or minimum expected adverse effects on normal cells, because normal cells rely on mitochondria, so it helps to activate mitochondria. But the disadvantage is the inhomogeneity in cancer or in tumors. Looking at, or this is a big problem in actually in cancer biology and cancer treatments. Looking at cancer as a homogeneous sort of cells is a big mistake, because cancer is inhomogeneous. There are cells that are proliferating. There are some cells that are in the necrotic cores. There are other cells that are close or nearby, or some stem cells, some cancer stem cells. All of these subpopulations of cells, they rely on different pathways. So you may be able to kill, for example, most of your tumor. However, you probably would end up activating cancer cells, the stem cells, the cancer stem cells. So we will talk in the rest of this presentation on the metabolic blackout strategy. But the problem of the metabolic strategy, again, is that it can cause some adverse effects because of the targeting or expected targeting of normal cells. It could be very effective in killing everything, but also it would target normal cells. So the alternative strategy that we decided to do is to essentially to use metabolic blackout strategy while loading our drugs in a specific delivery nano shuttle. So the idea is to try to find a specific receptor that's overexpressed in cancer cells specifically, and you try to prepare a nano formulation with a recognition terminus that would essentially recognize cancer cells more effectively than normal cells. And then you load these nano structures with your drugs and try to see what happens to and try to see what happens. What we did is we loaded a specific drug. With these data are essentially now in final stages of publications, and we saw that these molecules could effectively inhibit glycolysis very, very effectively. And as you can see, with very low doses, these doses are sub-micromolar doses. The IC50 is about 500 nanomole. And the same formulation, the same drug was also able to inhibit mitochondria, as you can see from the oxygen consumption rate. It's actually inhibited completely also, and the concentrations are very low. This is sub-micromolar. And we studied whether these would lead to effective cell death. And as you can see, if you compare the free drug with with the nano formulation, you can see that this is in nanomole and this is in micromole, that the effective dose is essentially reached sub-micromolar range, which is really very, very promising. You can see that treatment for a very short time with these drugs can kill completely your cancer cells. And as you can see, when we studied the mechanism of cell death, we found that the mechanism of cell death, this is the formulation, the nano formulation. After about 24 hours, all cells really disappeared, while in the free drug, you can see that the cells are still there, and there are also signs of apoptotic. The mechanism is apoptotic cell death through the PI and annexin V staining. So I will just move forward. We also made sure that this cell death or induced cell death is minimally through necrotic pathways. We also proved that the apoptosis played very important role in this by following caspases, and you can see that the caspase level is the unactivated level is decreased to the account of the activated level by these treatments, and very, very effectively by the nano formulation. We also studied the cell growth or the growth arrest by these formulations, and as you can see, there is a growth arrest in the G0, G1 phase of the cell. And also we studied the mechanism where we followed the reactive oxygen species and transmembrane potential of these cells, and you can see that the treatment with the nano formulation led to an increase, a very large increase in the staining of a reactive oxygen species sensor dyes. But this is the cells that are treated, and you can see that the intensity of the staining is very, very high due to elevated reactive oxygen species, and also due to the hyperpolarization in mitochondria. We studied the different metabolic complexes in mitochondria, and we could see that, for our surprise, that this formulation targets also mitochondria. So now the questions, are we specific or not? So it's just to give you an idea.
We have run out of time, Professor Sam.
Sure. To give you an idea, actually, we also discovered that this formulation targets mitochondria specifically, and we compared with normal cells. This is an embryonic kidney cells, and you can see from the staining here that our drugs, they actually stain mitochondria specifically, but it's lacking in the normal cells. And we could also find that this formulation induced cell death in cancer cells 200% more effectively than in normal cells. We are targeting several pathways. I'm sorry, I'm running out of time. And the conclusions is that probably we're hoping that we are providing a sort of new strategy for treating cancer cells while sparing normal cells. And I would close by saying that, of course, this is only the first step. Cancer is very, very complex, and if biology was so simple that we could understand it, we would be so simple that we couldn't. Thank you so much for your attention.
Thank you, Professor Sameh. I'm sorry, I will not allow any question because we have completely run out of time, but the professors are here at your disposal in networking.
Right, it's just, I'm sorry, I have to acknowledge the students that really did this work. Ahmed Abdrabbo and Asma Reda and Abdurrahman and my group, and also the support of Zuhail City. Thank you so much, and I'm sorry for being late.
Yeah. Thank you. Now, I was asked by the organizer to please fill in the forms that are in your bag on the feedback. There are 2 forms in your bags for feedback, and you give it to the organizer. And I have to acknowledge that we have had 3 outstanding talks of superb international level. Thank you, and I thank everybody for listening in.